All right, today we're going to start talking about active transport. Now, active transport, here are a couple examples. We're talking about two different types of ion pumps. Now, an ion, you guys probably don't know, maybe you do. Anybody know what an ion is? What's an ion? Okay, it's a molecule or an atom that has a charge. So it basically, we'll say ions, anything with a positive or negative charge to it. All right. Sodium potassium pump is an ion pump. Technically, a proton pump is also an ion pump because it pumps hydrogen ions, H plus ions, across the membrane. But we'll talk about what those two are in this section. But in active transport, we have the cell spending energy, using ATPs to move molecules across the cell membrane. So that's the key of active transport. So let's first talk about what's actually going on. In active transport, molecules are being moved against their concentration gradient. So if we take our analogy from yesterday of the ball rolling down the hill, this is now we're pushing the ball up the hill. Okay, to push that ball up the hill, that requires energy of you. You guys are a very chatty group today. Okay? So to get that ball up the hill, we have to expend energy. Okay, that's active transport. Because the bottom of the hill is the low concentration, top of the hill is the high concentration. That requires us to spend some energy to get that ball to move against the way it naturally wants to move. So we move from low to high concentration, involves transport proteins. Okay, these transport proteins are different, though, than the carrier proteins from facilitated diffusion because these transport proteins require energy for them to do their job. They pump molecules across the membrane. This energy comes from, again, ATP, which is adenosine triphosphate. So here's a picture of a generalized ion pump. Well, here's the protein embedded in the membrane. So this is the pump right here. It's the carrier protein. The ions will fit in that carrier protein. Now, carrier proteins are specific to the ions that they can transport. So let's say this might be sodium. Well, this carrier protein only works on sodium ions then. It won't work on potassium or calcium or hydrogen. It only works on a specific type of ion. But just bonding to the protein pump doesn't do anything. To get this protein to change shape, we have to add energy. When we give it energy, it changes shape, and then that moves the molecule across the membrane. What I want you to notice, though, is that in this drawing, we are moving from a low concentration of ions to a high concentration of ions. So this means this is something the cell wants to either have a lot of inside of it, so I have a high concentration of whatever ion it is inside, or if we reverse this, if this was the outside and this was the inside, this is something the cell wants to keep out, wants to prevent from coming back in by pumping molecules actively out of the cell. So let's look at the sodium potassium pump. It's a specific example of this ion pump. So this sodium potassium pump maintains a certain ion concentration inside of cells. Now, animal cells will pump sodium ions out into the cytoplasm, while at the same time pumping potassium ions back in. This is kind of a two-way pump. It pumps certain molecules out, pumps other molecules in, or I should say ions. The result is a high concentration of potassium inside the cell and a relatively low concentration outside, and then a high concentration of sodium ions outside the cell, but a low concentration inside. So it's kind of the reversing the directions of these ions. I think a picture is going to help you understand this a little bit better. Now, what's interesting about the sodium potassium pump is that it has 3 spaces to fit 3 sodium ions. That's what Na plus represents. Energy is added, pumps those ions out, but that gives a change in the protein's shape. That allows 2 potassium to fit, The enzyme goes back to its original shape, and 2 potassium get pumped back in. Now, these have charges. So essentially, we pumped 3 positive charges out, but only pumped 2 positive charges back in. What does that do to the 2 different sides of the membrane if we keep going with this 3 out, 2 in, 3 out, 2 in scenario? What starts to happen to the outside and inside of the cell? Kevin?
The inside becomes negatively charged.
In comparison to the outside, it's negative. Okay? The outside has more positive charges, so think of it as being more positive. Well, if this is more positive, well, then this is more negative. And that creates what's called a potential difference across the cell membrane. And this is how things like action potentials, how your nerves work. When you get a nerve impulse that runs down your body or runs down from your brain to your fingertips and stuff like that, it's all because of this difference in electrical potential. Yeah?
Isn't that also how a battery works? It has a positive ion.
Exactly. It's exactly like a battery. And when you connect the 2 sides, the electrons want to flow.
The voltage is measuring the potential difference.
Yep. Same thing happens in your cells. We get a voltage across your cell membranes in your nerve cells. Was there another hand up? I thought I said somebody else's hand up. Okay. All right, this is just going to be a video of it.
High-throughput screening and high-throughput phenotyping have become key technology.
We don't want
The
ads.
sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel, that in turn drives the ions through the channel. 1 phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
So again, the cell needs to spend energy to get this movement to happen. So ion pumps, why are they important? Well, muscle contractions. Your muscles have specialized ion pumps. They actually pump calcium into a special holding container. And so when your muscles need to contract, it basically releases all this calcium, and that causes your muscle to actually contract. Right? And so we have to actively pump this calcium, though, into these little holding containers so your muscles can undergo muscle contractions. Nerve impulses, talked about the sodium-potassium pump. That's what's the idea behind a nerve impulse. Creates that potential difference, that voltage across the cell membrane so electrical signals can fire through those cells. And then absorption of nutrients, simply just getting molecules into your cells. Okay, when you digest food, you have a high concentration at first, but as that concentration goes down, if we didn't have active transport, you would reach equilibrium and you'd still have a lot of nutrients still in your digestive system. Okay, there'd be the same amount on each side of the cell, but we want to keep taking those molecules in so we can store them, and so we have to actively transport them into our cells. Even plants use active transport in a lot of ways. Nutrient absorption from their roots. Nutrients are more concentrated in the roots than in the surrounding soil itself, but plants are constantly trying to pull more nutrients in. If they didn't, the nutrients would diffuse back out, and the roots would lose the nutrients that they've been working so hard to pump in. Okay, so plants have to use energy to actively transport nutrients like magnesium and stuff and nitrogen compounds from the soil. Here's another example. This is a proton pump. We'll talk more about proton pumps in another unit, but it's nice to see at least an image of them right now. It's another type of ion pump that uses energy. Of course, we're going to add.
Although we generally think of movement protons across the membrane as a mechanism for driving the synthesis of ATP in chemiosmosis, protons can also be actively transported across the membrane at the expense of ATP. For example, a proton pump can be used to maintain a 100-fold gradient between the inside of a lysosome and the cytoplasm of the cell. The transmembrane protein that acts as a proton pump binds a proton on the inside of the cell membrane while it is in conformation A. The energy from ATP then drives the transport of the proton to the outside of the cell through the formation of conformation B. Release of the proton on the outside of the cell results in the protein reassuming conformation A, in which the next proton can attach on the inside of the cell and be transported.
Go the other way?
I guess it could, but I don't think it does. I mean, usually proton pumps are one directional. We're pumping them. I mean, that protein's not going to flip in the membrane, most likely. So keep pumping them into, what in this case, the lysosome. So here's kind of a review of all the different types of transport we've talked about. We have simple diffusion. Molecules can easily pass through the cell membrane, like oxygen, which is small enough to fit through the cracks. So oxygen, carbon dioxide, all easily fit through the cell membrane. Now, it's been recently studied that water actually has kind of a hard time getting through, because water, like if these are water molecules, has a slight charge to it, so they kind of get through slowly. But cells have these special channel proteins called aquaporins that basically kind of shield them from the phospholipid bilayer, the nonpolar region. So I guess you can call it facilitated diffusion, simple diffusion, doesn't really matter. Regardless, it doesn't use energy. But then we have bigger molecules, or molecules of charge, like let's say we have glucose. Can be transported with a facilitated diffusion. This protein does not require energy. Basically, when the molecule attaches to it, it changes shape and pumps the molecule in. All of these move from a high concentration to a low concentration area. Active transport is when you get the addition of energy and you're moving from a low concentration to a high concentration. You're moving against the concentration gradient. That does involve a carrier protein. That protein requires some energy, and that's active transport. All right, quick little brain break. I want you to create a drawing in your notes of one of the types of transports that we discussed. And then I want you to share it with someone sitting next to you and see if they can figure out just on the drawing. So don't label anything. Just draw it itself. See if they can figure out what type of transport you drew. So, the next type of movement is another form of active transport. But this is called bulk movement. When you hear the word bulk, like you buy items in bulk, what does that mean?
You buy a lot.
You buy a lot of it. Okay, so this is the movement of like big things, a lot of things, big molecules. Okay, it's a process where transport of very large molecules, such as big polysaccharides. These are like basically large sugar molecules like starch or glycogen are moved this way, or big proteins that are assembled inside the cell and need to be shipped out will move through bulk movement. Okay, and this can move with or against concentration gradients. It doesn't really matter about the concentration gradient because the molecule is so big. It has to be moved out in a different manner. It can't use a transport protein for this. But they're used by these membrane-bound sacs or pouches. They're kind of packaged up. And then sent in or out of the cell. We have two types of bulk movement. We have exocytosis and endocytosis. What do you think the prefix exo, not gossip girl? To exit. Okay, that's how material moves out of the cell. And then what about endo? To enter. It's how things move into the cell. There's two types of endocytosis. We have phagocytosis and we have pinocytosis. Phagocytosis, the prefix phago, means like eating. This is actually the most primitive form of eating. Cells actually engulf some kind of food molecule or other organism. You have specialized white blood cells in your body called phagocytes that seek out bacteria and viruses and fungi, and they basically consume the cells, those foreign cells, and they destroy them for you. All right? And then pinocytosis, this is referred to as like cell drinking, bringing in something like more solution-like, so some kind of dissolved solute, bringing that into the cell, or I guess releasing it from the cell. I always think of like Pinot, like it's a type of wine. Pinot Noir is a type of wine. And that's, or a pina colada, right, there you go. All right, that's just a liquid, okay, something more liquid-based. So let's start with exocytosis. Now, cell products are packaged by a special organelle in your cell called the Golgi Apparatus, or sometimes referred to as Golgi bodies. They package the things up in these little tiny vesicles, these little compartments called vesicles. So think of the Golgi as kind of like, you know, the UPS facility, and the vesicles like little tiny UPS trucks carrying the packages away from the facility. Now, waste products, things that are broken down in your cell that aren't going to be used, that are considered waste, those will be packaged into vesicles as well and shipped out of the cell too. The vesicles will fuse with the cell membrane and they kind of release their contents out of the cell. An example of this is when you cry, you have tear glands. You have specialized cells in your tear glands that produce a salty protein solution. Okay? What do you think that salty protein solution is for? Like, why would your eyes produce that? Aiden?
Stop them from drying out.
One, stop them from drying out. But why else? Why can't it just be water? Why can't we just secrete water in our eyes?
Because we need water.
Well, we need water, but the solution has water in it. So it's not just plain water.
This is a gas, but does it kill bacteria?
Yeah, it kind of keeps them clean, disinfecting. Okay? Now, how many of you in here wear contacts? Okay, you take your contacts out at night to sleep. Hopefully you don't sleep in them.
I do. Because I wear night contacts.
Oh, some people would sleep in them. Okay. Well, when you take them out, when you put them in that little container, you put some contact solution in it. What does that do?
Cleans them.
Cleans it. What do you think it's cleaning off of them?
Proteins.
The proteins from your tears. Okay? So that's why you have to use that contact solution, because it's supposed to clean off the protein that's have built up. If you're waking up with that goop in your eye, like that white, yellowish goop, some of that is the proteins that just have built up. My mom always called them sleepies.
Eye boogers.
We call them eye boogers too. Okay? That's from that solution that's being secreted by your eyes. Yeah.
Is it true that when you cry, some of it travels down in like this part and it comes out as snot, and that's why you have a runny nose when you cry? Because I learned that last year.
I don't think so.
No,
I don't think so.
No one's bad at teaching something.
I'd have to look it up though, but I don't think so. This, look, you learned something different today. So, more about the Golgi. The Golgi is a series of these flattened, like membrane-bound sacs. If you could see them under the microscope, which we can't, we don't have the kind of microscopes needed to see these. I always think, from the pictures I look at, from like scanning electron microscopes, really powerful ones, it looks like a stack of pancakes, basically. But what it does is what's important. It sorts, modifies, and repackages proteins for export. Stuff that's going to be leaving the cell, the Golgi apparatus is kind of putting a label on it and sending it on its way. All right, that's the function of the Golgi. So I'm going to show you this little video that will help you remember the function of the Golgi. This will get stuck in your head for the rest of the day. I don't know who Professor O'Brien is. I'd rather have this song stuck in my head than the original. What's the original song? All right. We're going to end it there. You can watch the rest. I mean, it is on my notes, the full thing. It's 3 minutes long, though. We don't need to watch the full 3 minutes. All right, but I do want to show you a other video of more pertaining to the actual function of it. Not as exciting, but
In eukaryotic cells, folded stacks of membranes called Golgi complexes serve as packaging and processing centers. Proteins for export travel from the rough endoplasmic reticulum to a Golgi complex, where they are channeled into sacs that bud off from the Golgi membrane. In the hormone-producing glands,
this is how the vesicles kind of
the
fuse the
sacs
cell
fuse
membrane.
with the plasma membrane, releasing their cargo into the bloodstream.
Blood?
That's your blood.
Is it, I'm guessing that's in slow motion though?
No,
Oh, it was only traveling one direction. Wait.
your blood does travel one direction.
Yeah, but doesn't it like slow down? It's like moving speed.
Not really.
Speeds up and then it goes
Not really. All right. Now a vesicle, is a membrane enclosed sac. So it's a little pouch basically. And their job is to store or to transport different kinds of molecules. One specific vesicle, vesicles are usually categorized by what they contain. All right, so vesicles that contain digestive enzymes, things that will break down big molecules, are called lysosomes. They're sac-like organelles. They come from the Golgi. The Golgi sorts out the proteins that are going to be used for, basically digestion. So it sorts out all the digestive enzymes, puts it into a vesicle, and that vesicle becomes a lysosome inside the cell. They basically break down big carbohydrates, big proteins, big lipids. They break down all those big molecules. They also, though, digest old organelles. Organelles that wear out, that no longer function, will be broken down and kind of replenished, or digested, I should say, by a lysosome. Now, there are other kinds of vesicles, like peroxisomes. There's another example of one that's involved in detoxification. We're just not going to talk about all of them. So let me show you a video of lysosome.
You guys are left.
Protists and other phagocytic cells incorporate their food into membranous pouches
like
called
tiny
food
feet.
vacuoles.
Again, this is the oldest form of feeding.
When digestion is required,
For a
the Golgi
digestive
complex
system, everyone.
sorts
These are
and
single-cell
pinches off
organisms.
membranous sacs filled with digestive enzymes called primary lysosomes. Primary lysosomes merge with the vacuole membrane, dumping in their digestive enzymes. The food vacuole now becomes the cell's digestive system.
So the food vacuole contains the food, and the lysosomes fuse with it, dumping the enzymes in. It makes this like big soup of enzymes and broken down food that will nourish these cells. This is actually a type of endocytosis right here. We haven't talked about it yet, but this is phagocytosis. Taking in something solid, this protist is taking in some bacteria, and it's going to digest it.
Eating crap. It's pretty good.
So there's the food vacuole, has a bacteria cell in it. Lysosomes dump off their enzymes, and they start to break down all the big molecules. So break down the proteins, the fats, the carbs, all get broken down by enzymes. Basically chopping it into little pieces, and then the cell can use those pieces to build other molecules.
As digestion proceeds, nutrients are pumped out, creating a stockpile of molecules for cell use and export.
How does it move?
Through cilia. We'll talk about those later in the
Lysosomes
year.
function as the digestive system for phagocytic cells. But in all cells, lysosomes break down cell organelles that wear out.
So this happens in your cells too. When organelles are lost their function or don't work anymore, they get broken down. All right, please put the Chromebooks away.